My name is Olivia Piacentini, and I will be conducting the DNA extraction lab. Deoxyribonucleic acid, or DNA, is a double-stranded helical molecule responsible for genetic information and passing down genetic information to future generations. In living organisms, DNA is involved in development and functioning of all cells. I will begin by gathering my split peas that have incubated overnight in the distilled water. I will use a spoon to transfer them to the mortar, allowing for as little distilled water to be involved in the transfer. I've collected a new clean drinking glass. I'm going to put half a cup of distilled water in with 2 salt packets. Now use a glass stirring rod to help mix until the salt is completely dissolved in the distilled water. Sodium chloride is added to neutralize DNA, break up protein chains, and help separate DNA from the mixture. Salt will neutralize the negative charge of DNA, making it less hydrophilic and less soluble in water. This will help the DNA strands stick together and form a precipitate. It will also help to break the protein chains that bind around nucleic acids, preventing them from being involved in the DNA precipitate. I will now spoon transfer the distilled water mixed with salt until the mortar is half full. I'll be using the pestle to crush the peas, and I will continue doing so until they're fully incorporated into the salt water. I will now put the cheesecloth over a new clean drinking glass. I will be using a rubber band to secure it. Now gently pour the contents of the mortar into the drinking glass. I will now remove the cheesecloth and the solids and place them in the trash. Now add in half the dropper of detergent into the solution with the DNA. Now that half of the detergent has been added, I will use the clean stirring rod to thoroughly mix the solution carefully. Detergent is added to lyse cells and release their contents, including DNA, into the solution. The detergent will break down the cell membrane and the nuclear envelope, allowing for cell contents such as DNA to be released. I'll place the cup with the DNA mixed with the liquid detergent into the fridge and set a 10-minute timer. I will now gently pour the cold DNA soap solution into the test tube until it's about a third full. I will now add half of the meat tenderizer into my test tube. I will now gently stir as to not break the DNA. I will now add the 91% ice cold isopropyl into the test tube at a 45-degree angle. Adding chilled alcohol to the sample helps precipitate DNA out of solution and removes impurities. Chilling the alcohol prevents the DNA from being denatured while purifying the DNA sample. The test tube is now upright, and I will follow and observe the solution for the next minute. Stringy white fragments are observed to be DNA.